So I had this camera, and I figured it'd be a lot easier and quicker to record than doing my phone. So we'll see how it works today.
Okay.
Yeah, we'll go ahead and start.
So does anybody have any questions on the exam? If you've looked over it any further? No questions? Okay. We talked about this last time, and I can't remember if I tested you all on these on the exam. Did I test you all on these topics? What's increasing the rate of reactions? Maybe I did. I don't know. However, we talked about 4 things that increase the rate of reactions. Okay? And the first one was the catalyst. Now, actually, let me back up for a second. We talked about reactions being things that occur whenever 2 molecules collide together. And then they either break, make, or
Any part.
or reform a bond. Yeah, right. But what increases rates of reactions is increasing the chances that 2 particles collide together. Okay, so whatever can increase the chance that 2 molecules can collide together is what typically increases the rate of reactions. I mentioned to you all, I'm not switching. I mentioned to you all, the first one was catalysts or enzymes. Okay, these were protein molecules, so large proteins that these were protein molecules, and I didn't show you all this, but I want to get the idea in your head. These are proteins that will take 2 molecules and hold them together in the same place. And that's what allows the reaction to occur. And in essence, if you're taking 2 molecules, we'll say 2 water molecules, okay, and they're stuck here. Obviously, this isn't a legitimate reaction, but they push them, they hold them there in the protein. And that's increasing the chances that these 2 particles collide together. Now, that just so happens to lower the amount of energy it requires for the reaction to occur. Okay? This is how catalysts work. But these other 3 factors are doing the same thing, just that they're free in solution. Okay? So you increase the temperature, the hotter the solution is, it increases the movement of particles, and therefore increases the chances that they collide together and have a chemical reaction. Okay? The concentration, if you increase the amount of substance in the solution, obviously you're going to increase the chances that those substances will collide together. And then the particle size, the smaller a molecule is, the faster that it moves, and so that increases the chances that they'll collide together. And these are things that increase will increase the rate of the reaction, but they're really at the very foundation is increasing the chances that 2 molecules will collide together, and then you have a reaction. So these 4 things are important to note, and these 3 factors are not the last time you'll see this, because this applies to a lot of other concepts that we'll learn about. Okay. Okay, so we finished up the reactions, and we didn't get to a lot of what we were supposed to cover on exam 1, but that's okay. We'll get through what we can get through. And so we've talked a little bit about chemistry. Okay, in chemistry, you all understand that these are whole separate classes on themselves. It's just sort of an introduction to understand the concept because it's the foundation of what we're talking about. But then you move to something called biochemistry. Chemistry is more talking about the foundation of chemical principles and understanding rates of reaction and calculating a lot of different things. But you all understand that this applies directly to our body. And so that's where we move into biochemistry, where we're actually working with molecules that are in our body and specifically learning about those reactions that are occurring, really with molecules that are in our body. And so typically, chemicals fall into two categories. The first one's organic compounds. Okay? And organic compounds are found in all living things. They're made by all living things, and they always contain carbon. They always contain carbon. So a way to identify if a chemical is organic or not is looking if there's carbon there. And today we'll certainly get to why carbon is so important. But then you have inorganic compounds, which is everything else. Okay? And so you're thinking that includes water. The most abundant molecule in our body happens to be inorganic because it doesn't contain carbon. A lot of salts, acids, bases are generally inorganic compounds. But anything with carbon is organic. Now, don't get confused, organic versus inorganic. Both are essential to life. And you already know that with water being inorganic. You need both compounds. That would be a really good true-false question. Okay. Now, obviously, like I've already said, the most abundant inorganic molecule is water. And water is incredibly important because it has different characteristics associated with it that help us maintain life. Okay? And there's 5 really important properties of water that we'll talk each one. First is its high heat capacity. Second is a high heat of evaporation. Next is polar solvent properties, and you all can imagine what this is talking about. Reactivity, and then there's cushioning. Okay? So the first one is high heat capacity. And this, I have to use the PDF. It's supposed to have like animation, so I don't show you all everything on the slide. However, water is able to absorb large amounts of heat. This is the first characteristic, is its high heat capacity. It's able to absorb large amounts of heat. At the same time, it's also able to release large amounts of heat. And you all can imagine that this is essential for our temperature regulation. Okay? We've already talked about how temperature is being regulated in terms of the blood, but what's making the liquid portion of blood? It's water. Our body is over 70 or about 70% water. This is how we're regulating heat. Now, during physical exercise, and we've already talked about this, when you're converting energy, what happens? What happens when you're converting energy? You lose energy. And how do you lose it? And you lose it in the form of heat. What's going on with muscle contraction, and certainly we'll talk about this next exam, is that we're converting chemical energy in the form of the bond, and we're converting it to mechanical energy. And always during a chemical or why am I losing my train of thought? Always during an energy conversion, we're losing its heat. And this is what's occurring during a muscle contraction, is that we're losing it as heat. We're losing energy. And so that makes our body fairly hot. What would happen? I'm maybe exercising, or my muscles are contracting a lot. I'm doing a lot of energy conversions, meaning I'm losing energy as heat. Very inefficient process. What's happening? How do we regulate that? I wish I had my sticky notes. What's happening?
You're sweating.
Okay, you're sweating. But what's the first change? What's the first thing that occurs? I'm exercising. I'm losing it as heat. What's happening? Okay, heart rate might increase as a result, but there's something very, very important that we've talked about time and time again. Concept is that we're losing homeostasis. So what's the first thing that occurs?
Your body sensing.
Your body senses it. Okay? What happens after you sense it?
Sends it to the brain.
Sends it to the brain. Afferent signal. Brain makes a decision. It's too hot. Now I need to release some of that energy. How are we releasing it? We're using the high heat capacity of water. Our blood is absorbing this heat. And it's making our blood hot. That's increasing our body temperature. How do we release it? Our skin is moving blood to the surface so that we can release large amounts of heat. This is why water, one of these properties of water are incredibly important. Its high heat capacity allows us to absorb the heat that we're taking in, and that you all understand that the heat is very dangerous for our body because our chemical reactions cannot occur properly. These bonds become unstable, so we have to regulate our body temperature. Water's high heat capacity absorbs that heat from the muscle contractions, and then we release it by putting the blood to the surface of our skin because of the high heat capacity of water. At the same time, not only do we move it to our skin, but also we redistribute the heat. Say we're only exercising our arms or our legs. There's a lot of heat being accumulated here. Our circulation allows it to redistribute to the rest of the body. Okay, so we're taking energy that's lost in the form of heat at certain locations. Water's absorbing it, we're redistributing it, and at the same time we're pushing it to the surface of our skin so that it can be released. Then we have the high heat of vaporation. Or, I mean, I don't really like this statement. The way, I don't even know how I would rephrase this. But water has another ability. We talked about its ability to take in heat, but what do you all know when you boil something? What do you all know about if you make a liquid too hot? It evaporates. You lose it. Okay? If you're thinking about it, if we're creating lots and lots of heat, say I walk outside, it's 100 degrees and I start running. That should, in theory, start boiling my blood. I should start losing some heat. Or I should start losing some liquid. Well, the ability for water to not only absorb heat, but also not vaporize due to high amounts of heat that we're taking in. This is what's called the high heat of vaporation. I don't really like it. And you all understand that water has multiple forms. You have the liquid form. If you get too cold, you have the ice form. And if you get too hot, you start to vaporize. Okay? Well, this temperature that you reach before you get to vaporation is incredibly high. Right? This is 100 degrees Celsius. Our body's staying at 30, what's our body staying at? 37 degrees Celsius. That's an incredibly high heat or high point before you reach vaporation. Okay? So this keeps our water within our body without losing it. Do you all think it's hot in here? Like a little bit? I'm like sweating. Now that's really all I need you to know, and you all know how this applies to temperature regulation. We're taking in large amounts of heat, and we're not releasing the water, we're releasing the heat. Okay? And then there's polar solvent properties. And you all already know that when we're talking about polar bonds, we're forming two partial charges. We form a partial negative on an oxygen or another electronegative element. It could be nitrogen, but it has to be something that's incredibly electronegative with something that's a lot less electronegative in it. In terms of hydrogen bonds, it's always hydrogen. Okay, but this element can be different, oxygen, of course. But obviously, you all know we have a lot of water, so this is the most important property. However, we form these partial charges. Now, these partial charges obviously are called polar, but they allow us to do so many different things in our body. And one of the important things is forming these things called hydration layers. Now, I have here sodium chloride, and we'll talk about that in a second, but I really want to talk about this compound here. And how would you form this compound? How would you form something like this? We talked about it. I said you all didn't need to know the example, but if somebody remembers it, I'll certainly give you a sticky note. How would you form this compound here?
Synthesis.
Synthesis reaction, exactly. This happens to be a protein This happens to be a protein, and we'll talk a lot about it, but proteins are making up our entire body. Okay? Now, you all understand that these are chemicals. What's causing, and what happens when another chemical interacts with another chemical?
There's a reaction.
There's a reaction. So how do you stop something like this from reacting from something that it doesn't need to react with? We have charge-charge interactions. This is why water is so important, is because it will start forming, based on its negative and positive charges, what's called hydration layers. In essence, it clouds around the whole protein structure or whatever chemical you're talking about to prevent it from interacting with something it doesn't need to. Okay? And now you all know that we have all kinds of stuff that's floating through our blood. We have all kinds of proteins. We have all kinds of hormones, which a lot of them happen to be proteins. We have to prevent these things from interacting with other molecules when it doesn't need to. And one of the ways that we do that is we form these hydration layers through the polar property of water with negative charges and positive charges. And you don't need any other charges because that's all there is. Okay? So sometimes oxygen will interact with maybe a positive charge on these proteins. Sometimes with positive charges on hydrogen will interact with the negative charges on the protein. And that prevents it from interacting with other things. It hides the molecule from other interactions. Now, at the same time, this is how we often break ionic bonds. You all know ionic bonds are positives and negatives. Now, positives and negatives interacting with each other. The most common one happens to be table salt. Table salt is sodium chloride. Maybe I have a picture on the next. No, I don't. Salt happens to be sodium chloride. That's an ionic bond. It's an intermediately weak bond, you all remember that? And what happens when you put salt inside water? It dissolves, okay? How does it dissolve? It dissolves through these polar solvent properties of water. The positives on the hydrogens interact and form a hydration layer around the negative chlorides. And the negative charges on oxygen, of course, these are partial. So we're not forming permanent bonds here. Right? If you take the water away, eventually you'll see the salt turn back into grains or whatever you call crystals. However, the partial negatives of oxygen form a hydration layer around the positive sodiums. And so this is how they're dissociating in solution. Now, maybe some of you all know this, but if you take, for example, salt or sugar, it's the same process with sugar, and you start to pour it into a liquid. Most people do, maybe not salt, but certainly sugar with like sweet tea or iced tea or something. What do you think would increase the rate of the solution dissolving? Heat. So if it's too cold, if you put maybe sugar into your solution, you're not going to dissolve it as much because the reaction is not fast enough. You're decreasing the rate of these molecules moving or interacting with each other, and so then it takes a slower time for it to dissolve. And so, when you have like cold solutions, for example, everybody knows that you dissolve sugar in sweet tea, it does not necessarily dissolve because the molecules are not moving as quickly. And so it doesn't dissolve. These, don't get it twisted, these are still chemical reactions, right? It might not be a chemical reaction here because I just told you you're not sharing any electrons, but you all know in order to pull these two things apart, that's a chemical reaction. You're breaking a bond. And so these concepts still apply. And it's the same if you were dissolving salt and water. If you put it in a hot liquid, it will dissolve much, much quicker than dissolving it in a cold one, and for reasons you already know. The other one is reactivity. This one's not as important. The first three that we just talked about are really important, but water is the most inorganic molecule in our body. You already know that, and it's the main solvent in our body as well. And I've already been harping that to you all. Our blood, this is where a lot of things are dissolved in, but also in our cells, these are all liquids as well. So these are the solvents that are holding all the solutes that we know of. And any molecule that you can think of is the solute. And In our body, the solvent that's universal is typically water. You're talking about the cells, the cytosol or the cytoplasm, that's water. Okay, the blood is majorly water. The space in between our cells is majorly water. And so adding water to chemical reaction can result in many reactions occurring, especially these two, decomposition and hydrolysis reactions. And we'll talk about each one of these after this PowerPoint. However, why would water be so important with chemical reactions? Let's think which. If I took two molecules, two single molecules, and I put them on that table right here. I put two molecules right here. What's the definition of a chemical reaction? Breaking, making, or forming new bonds, but it's caused by two molecules interacting with each other. Okay, so let's think about this simple concept. Adding water to the reaction allows the molecules to move, right? If I drop these two things in this water, the chances of them interacting with each other is much higher than I just lay them here on this table, right? Much higher. If I just took two molecules and put them here, they're not going to move. But if I have water, I now have this great solvent where they can float and they can move, and now they're allowed to interact with each other. So there's another property of water that sort of goes along with reactivity, is it allows these reactions to occur. And then there's cushioning. And this is fairly important. Water is not compressible. Okay? You can't compress it. And this was a question on the exam, a true-false question. And this is essential for our body to protect us from physical trauma. Remember, we talked about those body membranes, and you had the visceral layer, you had the parietal layer that was protecting our organs from rubbing against each other and getting damaged. Well, at the same time, we also have a lot of forces that are hitting our body. We are constantly experiencing trauma to our body, and I'm not just talking about your heart beating. Okay? Whether you know it or not, we constantly have trauma to our body. Water has this property of not being compressible. And so especially when we're talking about compressing, we're removing the air. And so at some point when you push these molecules down, it won't go anymore, no matter how much force you give it. Now, I mentioned to you that your brain and your spinal cord sit in this type of body cavity as well. Right? And there was that cerebral spinal fluid that's flowing through. This was a body membrane. Right? But what's so important about that liquid that's flowing through here is that when we experience trauma, yeah, the skull might break, but if it doesn't, or the spine, one of the vertebrae might break, but if it doesn't, we are protecting our body from some sort of damage because water is not compressible. It will stop the force from pushing through the spinal cord or pushing to the brain. Now, again, like I said, you have the skull. So if the skull breaks, obviously use the compressibility. But when you're talking about a closed system, like in this case, it's closed. The brain is surrounded by a skull and vertebrae. The water is not compressible, and so it prevents us from being damaged. Okay, very important. And this just doesn't apply to the brain. The brain and the spinal cord are the most important things for our body functioning, and you already know that. So this is obviously the most important feature of it. But this applies to a lot of different things. Okay, so we've talked about salts a lot. We've talked about the cations and the anions, the positives and negatives interacting with each other. Salts are always formed by these two charges. Essentially, all salts are made through ionic bonds. Okay? But salts dissociate in water, and you know that because of the polar properties of water. And we'll talk about this too, is that when you have these individual elements that have positives and negatives on them, these are collectively called our electrolytes, and I want you all to know that. These are the electrolytes that are in our body. Sodium, calcium, potassium, chloride, I can't think of any more. Magnesium. These are all electrolytes that are formed through our body that are doing very important functions. But also, What's interesting about these charged particles is they conduct electrical currents. And what do I mean by that? we've already talked about electricity. Right, I told you that electricity is the simple movement of a charged molecule, specifically electrons, but it can be positively charged. It's the movement of charged particles. Did somebody say something? Oh, sorry. It's the specific movement of charged particles. That's what's generating electricity. Well, in our body, we have these ions that are constantly flowing. That's generating electricity. And so these electrolytes are very important for the electrical functions of our body because they're charged particles that are moving. Okay? And this gets very important, especially when we're talking about neurons, which are brain cells. These are how they're sending signals to the other parts of our body. Remember we said that they sense them and they send them to the brain. How are they sending a signal to the brain? They're not taking the temperature and moving it to the brain. They're sending a signal via an electrical charge. This is movement of those electrolytes. And we have millions of these things. And on top of that, our heart has millions of cells that need electricity to pump. That's why when you see on the EKG, you see those electrical charges that are moving. That's movement of ions. That's movement of these electrolytes. Chloride, sodium, calcium, which is not on here. But it's movement of these electrolytes that's generating an electrical current that allows things like our heart to beat, for our brain to send signals. And then, of course, this is something you'll learn next semester. The neuron will learn this semester. But in the kidney, movement of ions is also incredibly important. Okay. And then we have acid bases. I'm not gonna, further harp on this because, we touched on it in the lab, and I don't think it's necessarily important to go into detail about calculating certain things. But acids, there's a fact about it. They have a sour taste. Acids are what actually generates a sour taste in our mouth. Okay. Now, by definition, you already know that acids are high concentration of hydrogen, low pH, which makes it more acidic. Acids are also known as proton donors. And you remember I told you, and I'll keep reminding you of this, hydrogen is also called a proton. Okay, free hydrogen ions are called a proton. So that's where this proton donor concept comes from. They donate hydrogens. Now at the same time, bases have a bitter taste. That's just the fact about them. I would like you all to know that for the exam. But they increase or they decrease the amount of hydrogen ions that are in solution. They're also known as proton acceptors. And some of these common ones are hydroxide, which we've already seen. We've seen bicarbonate, HCO3-. And you all remember, I told you I would learn these element symbols because I'm not going to write them out anymore. NH3, nitrogen, hydrogen, 3 of them. Hydrogen, carbon, oxygen, oxygen, hydrogen. Okay? These are, and I do want you all to know these 3 things. Hydroxide, bicarbonate, and ammonia, NH3, are common bases. Now, what makes them bases? They have these negative charges associated with them that interact with free hydrogens or free protons and soak them up out of solution. Remember, acids are free hydrogen ions. They're not hydrogens. They have to be free in solution. And I won't talk about this. Okay. And then you have this pH scale. And I'm not going to make you all have to calculate pH whatsoever. I don't think that that's important for this class. You can get it in a chemistry class, but you all know more hydrogen ions is lower pH. Less hydrogen ions, higher pH. Okay, I'm going to leave that there. We'll go on to the next PowerPoint. Okay, does anybody have any questions so far? Pull this out into- Okay, so this is like the lecture 3, I think, on Blackboard. Okay, this is lecture 3 that's on Blackboard, and so, you know, I put this back in there just because it was from a different class, so I didn't take this slide out. However, we talked a little bit about inorganic compounds. That's what we just talked about. It was inorganic compounds. Water being the most important. We talked about each one of those properties. And then we talked about salts and then pH a little bit. Now, organic compounds. Organic compounds always contain carbon. It's very important. Now, why is carbon, out of all the molecules, so important? We've talked about carbon a lot in this class. We even talked about it when we were talking about isotopes. Carbon is a small atom and it's electroneutral. So it doesn't have any charge. And it always shares electrons. But not only does it always share electrons, it can form 4 bonds, which makes it sort of like a spider. It's unique in that it can form 4 bonds with molecules and it's not charged. So, in essence, carbon is able to form these large chains and ring structures. You remember I showed you that ball and stick model of carbon, and it looks something like this. Each one of these points were those big black balls, the carbons, here. Well, its ability to form 4 bonds, and why can it form 4 bonds? Why can it form 4 bonds? Why is carbon able, and why does it form 4 bonds? You can't get it.
Because that's not like the other electron.
Because of the valence shell. It has 4 in its valence shell. You won't see that here. There's 6 electrons. It has 4 in its valence shell. So it needs 4 to be stable. And it does that constantly. It does that constantly. And it allows it to form these rings or these branches of compounds. And not only does it allow it to form this, but it forms other bonds with other molecules that stick off of it. Okay? Now, oftentimes it's hydrogen, but there are many other molecules that will bond there. And so that's why carbon is so important. It's not too electronegative, it's not too electropositive. It's right there in the middle, electroneutral. It's got 4. So it needs 4 to become stable. And so it forms all of these bonds, and it makes up every single protein in our body. Now, organic compounds, like we've already talked about in lab, form 4 broad categories, and these are called macromolecules. Okay? And the macromolecules, which we've already talked about, are carbohydrates, lipids, nucleic acids, and proteins. Okay, these are very important. You already know that from lab. And this actually is kind of good that we talk about this today because you'll have a quiz on this today in lab. So, this will be good for you. But all organic compounds have these building blocks called monomers. You all remember I showed you that sort of brick, that large brick, and you have these different types of individual bricks, all these that make up these 4 different things. These are the walls. This is the whole brick wall, but the individual bricks are the monomers. Now, monomers grow to form what's called polymers, which are essentially the same building block, or multiple building blocks together. And then the macromolecules are here. So polymers are multiple monomers put together, and then those polymers can become macromolecules. But it's important to understand not all polymers are macromolecules. Okay, polymers is just saying that you have multiple monomers that have come together. The macromolecules are these four things, and only those four things. And so that's sort of explaining this here, but macromolecules are large, complex, with over 100 units associated with them. They're very large molecules. Again, all polymers are macromolecules, but not all macromolecules. Ooh, I have that back. I have that backwards. Don't pay attention to that. So how do you form these macromolecules? Macromolecules can be formed through two types of reactions. One of them is a dehydration reaction, and another one's it can be one of the reactions is dehydration where you're forming them, and the other one's called a hydrolysis where you're breaking the bond. Okay, so we have dehydration and we have hydrolysis reactions. Dehydration, as it sort of states, is during the reaction, water is lost. So you have two monomers here, and they each, and we'll see large structures associated with them. This is trying to get the concept, is that usually you have two hydroxide ions, okay, or two OHs associated with these monomers, okay, and these monomers could be glucose, it could be sugar, okay. They lose water, and you can see where it's losing water now. One of the molecules loses one hydrogen, the other one loses a full hydroxide, and you lose water and form a covalent linkage between the two monomers. And this is how we're building them up. It's through a dehydration synthesis reaction. So this is still a synthesis reaction, it's just got another name to it called dehydration because we're losing water during the process. Okay. The second type is called a hydrolysis reaction, and we'll see each one of these reactions many times throughout this course and throughout next course. But water is required to do the reverse reaction, to break the bond. Okay, so say for example you start storing up glucose. What would cause the storing up of glucose? Say I eat something. Insulin. So insulin would be released. Then we pull the glucose out of the blood and we start to store it up. We don't store it up as single units like these individual monomers. We bond them together to make them, to store them. Well, to break them when we need it, like we haven't eaten, we use these hydrolysis reactions. We add water to the system, and that then breaks these bonds. Okay, so know these two reactions. Just know one's adding water. I would like you to know that there are hydroxides or OHs that are at the ends of these molecules where they're bonding at, the interaction between the two. Now, this is more, this is more to help you all understand as we're moving through this lecture, is that these compounds are written in a unique way, and they're written, different depending on, the type. There's different names for these types of structures. However, this is the most common type of writing for the structure, is that you will see that this is obviously a chemical compound, and here we have written all the elements. Well, it gets very tedious when you're talking about carbon because it's making up all the organic compounds in our body. So they abbreviate them as these points, where these two lines form that little V. That indicates that there's a carbon there. Now at the same time, these indicate a carbon, or these would indicate a carbon as well, unless something is specifically written there. So any point that you see on these lines indicates that there's a carbon there, unless I write it. So in this case, you see that oxygen is written here. There's an oxygen there instead of a carbon. Okay. Now, if that oxygen was erased and made that little V, that's a carbon. And the same here. These two structures aren't the same. However, and the reason is because this line has nothing associated with it. So you assume that it's a carbon. In this case, I've written the oxygen there, and so the carbon's not there. Okay? And I would like you all to understand how to read these structures, because I'll give you structures like this constantly. And in the case of carbohydrates, some of them you'll have to write in where oxygens are. And you'll have to understand that there's carbons in other places. And you know, I'll let you all know that throughout this lecture. But again, there's a line. If there's any point, you assume it's a carbon. There's a carbon here, there's a carbon there, there's a carbon here, there's a carbon here, 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 unless it's written. Okay. Okay. And then here's the first one that we're looking at. These are carbohydrates. And hopefully, you know, a lot of this is review, but the importance of carbohydrates is that this is an easy use of energy for our body. Again, there's that word energy again. You all should understand, energy is the movement of charged particles, specifically electrons. They contain hydrogen, oxygen, and carbon. You can see that here. And this is a fairly large compound, and they make up 1 to 2% of cell's mass. An important thing that you have to know about carbohydrates is they have a 2 to 1 hydrogen to carbon ratio. In essence, for every 1 carbon, you have 2 hydrogens in any compound that you're talking about. Okay? And you can modify carbohydrates to form these other things called monosaccharides, disaccharides, or polysaccharides. Okay? So this is an individual glucose. This is a monomer. In this case, when we're talking about carbohydrates, it's a monosaccharide. Okay? Now, if I took 2 glucoses together and put them together, you form what's called a disaccharide, and then you start going beyond that, you form a polysaccharide. So there's 3 categories associated with them, and the first one is monosaccharides. Okay? And there's multiple types. These are defined as the simple sugars, and they're monomers. Okay? There's 5 different types, and there's a little bit more, but these are the 5 main ones that you need to know. They contain a 1 to 2 to 1 carbon to hydrogen to oxygen ratio. Okay? So I could give you, let's say, carbon, hydrogen, oxygen. I could say that there's 3 carbons in this compound. Or let's say, let's say that there's 2. We'll say that there's 2 carbons. We'll make this is fairly easy. How many hydrogens would there be? Huh? So if it's a 1 to 2 to 1 carbon to hydrogen to oxygen ratio, and I have 2 carbons here,
4.
would be 4. And then how many oxygens? 2. It's as simple as that, and I will certainly give you something like that on the exam, and the final exam will have something like that as well. This is the chemical ratio that makes up monosaccharides. Okay, and so we can write their general formula as CH2O. Okay, because for every 1 carbon, there's 2 hydrogens, there's 1 oxygen. So this is the general formula. And then you can add a little subscript, which indicates how many of these monosaccharides you have. Okay, so if I put maybe 3 down here, I have 3 monosaccharides together. And so then you can, multiply 3 to oxygen, you can put a little 3 down here, multiply 3 by hydrogen, you can put a little 6 here, and you can put 3 to carbon, you can put a 3 there. Okay, so this is indicating how many of this compound that you have. Now, these compounds are named based on the number of carbons that they have. And this is more of a general name. Obviously, we have these, more, unique names associated with some of these compounds that are heavily present in our body. But we also have general names for them based on the number of carbons. Okay, so a glucose is a hexose sugar. It has 6 carbons associated with it. Hex indicates 6. and maybe I'll come back to these prefixes now. These are the prefixes for carbons. Okay, so you put these before the name starts. You have meth, which indicates 1, F, which indicates 2, probe indicates 3, but 4, pent 5, hex 6, hep 7, oct 8, non 9, and dec 10. Okay. These, I would like you all to know these prefixes. Especially 5, 6, and then 1 and 2, meth and F. Okay, so I will test you on these two and 5, 6, and 7. Okay, but I would like for you all to know all of them. But I'm just letting you know right now, I will certainly test you on those. Now, when I'm looking at glucose, I see that there's 1, 2, 3, 4, 5, 6 carbons. Okay, so I named that a hexose sugar. Now, if it has 5, which is also fairly common, you have 1, 2, 3, 4, 5, 6. Why am I counting? 1, 2, 3, 4, 5. That's it. Oh, this is a 6. Oh, sorry. These are hexoses. Each one of these have 6 carbons associated with them. But then you move to the other ones, which are pentose, which have 5. And then you can count 1, 2, 3, 4, 5. And this is deoxyribose, this is ribose. We'll talk about that in a second. But we can name these compounds generally as a hexose sugar. And if I say hexose sugar, I could be talking about any one of these. Or I could say pentose sugar, and I'm talking about any one of these. Now that's important to know because these are not the only 5. Okay, there's many different types. These are the most common, of course. And then pentose, yeah, of course, we're talking about 5. There's this concept, and I didn't really go into it a lot last year, but I do want to get this point across, is that you can change the way that these molecules look. You can change the interactions between elements, okay? I can put, for example, an oxygen here and move a carbon here. Or I can, in this case, I could maybe flip some of these components. Like I could take hydrogen and hydroxide and flip it. Okay? You can change the structure of these chemicals but maintain the same elements. That gives them different properties. Okay? And whenever you have compounds like these that have the same elements, in essence, I write the same chemical formula for each one of these things, but they look different. The way that these bonds are formed are different. It's called an isomer. Okay? And so I would like you to know that these are all isomers of each other and what that means. They're the same chemical formula, but they just look a little different. Where their bonds are located for different elements is different. That changes the way that they interact with other things. I'm trying to, yeah, we'll talk about ribose and deoxy. Well, actually, I'll introduce this now, since I have a story to it. Is that you have these two compounds. You have something called deoxyribose, and you have ribose. What's important about these things is this is what's making our DNA. Deoxyribose is what's making the foundation of our DNA, and we'll go into that because it's one of those polymers or those why am I losing my train of thoughts on this? Oh, macromolecules, sorry. And so we'll talk about it more in detail, but I want to introduce the structure of it because you'll have to identify it on an exam. Is that deoxyribose is a pentose sugar. It has this structure associated with it, but there's a difference right here. where at this carbon you have a hydrogen here. You've lost an oxygen. That's what makes it deoxy. In this case, when you have a ribose, you have the oxygen present on this carbon. Okay, that gives it a very unique property that allows these two compounds to interact or to form different chemical reactions. Okay, don't ever lose sight of that. When we're talking about the difference between all of these, I'm not trying to make some sort of imaginary thing that you can't see that you have to memorize. These structures give them very different properties that allow them to interact differently with other compounds. Okay? Now those interactions you don't have to know, but you will learn them eventually one day. But these differences in structure allow them to interact with other things differently. Okay, so I'll tell you right now, I will give you this figure and I will remove these two things and ask you to identify or draw on there me a deoxyribose. Now I might give the rest of the structure to you, but you would have to determine what's making it deoxyribose versus ribose. Okay, so know that structure in detail. Okay, then you have the disaccharides. I mentioned to you disaccharides are multiple monomers put together, and it's two because it's disaccharide. But what is the reaction that's forming these things? What's the molecule that would form a disaccharide? I have two monomers. How would I form it? Who said that? What? No, what would be the reaction that forms these two together? So I have single monomers. How do I bond them together?
Synthesis.
Okay, a synthesis reaction, but more specifically what?
Hydrolysis.
No, hydrolysis is breaking. Dehydration. So don't forget, we're talking about these forming these molecules. We had dehydration. We're losing water and we're bonding that oxygen together. You all just saw that image on this molecule. If these two things were separated, I have a hydroxide sitting right here, just like this, and I have a hydroxide sitting right here. We bond the oxygen together and we lose water during the process. That was dehydration. Now, if I wanted to break the bond, then I would just add water. And then one of those hydrogens will bond to this hydroxide and make this one hydroxide, and then that other oxygen and water on the H2O will just bond right here and separate the two molecules. That was hydrolysis reaction. So disaccharides are incredibly important in our body, okay? Because I keep doing this, I keep going back, but a lot of times people lose the big picture of what's going on. Where did these things come from? Where did these carbohydrates, where did these proteins, where did all where did these macromolecules come from? They came from the food. We have to eat it. We have bread, we have beef, or whatever you want to say, and we take these large compounds of food and we eat them. And as we move through the digestive tract, that single tube, we are breaking it down into smaller and smaller and smaller and smaller components, and then we absorb it into the blood. And then in order for us to absorb it, we have to break some of these things down so we can get the individual glucose molecule. We can get the individual fructose molecule or the galactose. But they form these disaccharides first, and we have to cleave these bonds. Okay? How do we do that? How do we cleave these bonds? What's that called? Now we just talked about the reaction, but let's think big picture. What are those? How do we break those off? What is that? That's a chemical reaction. And then we absorb it into the body. And we'll talk about those processes and how we're absorbing it. But oftentimes they require these. Oftentimes they require these enzymes, and we'll talk specifically about lactose, how we're breaking that down. But a lot of these things require enzymes that break these bonds. Now, I want you all to know there are multiple different types of disaccharides. We have sucrose, maltose, and lactose. Now, these multiple things are made up of different monomers bonded together. In the case of sucrose, that's glucose and fructose. In the case of maltose, that's glucose and glucose. In the case of lactose, that's galactose and glucose. So you all can see right now that glucose comes in many different forms in our body. We're getting sugar in multiple different forms. Okay, in these forms of disaccharides, and there's one more we'll talk about, but never lose sight of the big picture. We are trying to get these individual molecules into our cells so that we can make energy. Okay, so again, you all generally have to know these structures and identify them, but in this case for the disaccharides, I just want you to know what's making them up. Don't memorize these compounds. Just memorize the names of them and what disaccharides are making up. Okay, and then we have polysaccharides. And polysaccharides are links, mainly you could say they're links of disaccharides, or you could say they're links of monomers, but they make these large compounds, hundreds and hundreds and hundreds of molecules bound together, or monomers bound together. And they're used for storage. We have to have a way to store these things, right? I told you all that glucose was incredibly toxic to our body. That's why we have a very powerful mechanism to pull it out of the blood. Now we have to have a way to store it. We store it in a way of what's called polysaccharides. You all just seen that those are hundreds of molecules bonded together. The main one is called glycogen. And you have to know this. Glycogen is the primary source of glucose. These are hundreds of glucose molecules bound together that are again made through synthesis reactions, made by a dehydration reaction, and we store them in the cells. Glycogen is the primary storage of glucose. So you all know now that when we're taking in insulin, we are pulling it into the cells and we're forming this compound glycogen. We're simply pulling it out of the blood and into the cells, and we're bonding it onto this huge branching polysaccharide to store it. And then when we need it, right, you had that hormone glucose or glycogen. And then glycogen is releasing maybe an enzyme that comes here and starts cutting these bonds and releasing the glucose into the blood via hydrolysis reaction. Okay, there's another compound called starch, which is the same thing. There's just a different bond associated with them. Okay, starch is only glucose, and it's differing in the branch points, the bonds that are connecting them together. So it looks a little different. Okay, and it's only found in only found in plants, and then you have cellulose, which is also found in plants. The same thing. Okay, so we eat a lot of starch in our diet, and we have you won't find glycogen in the food. Usually you'll find it in other forms because this is how we're storing it. However, cellulose, we won't eat it because we cannot digest it. And the reason we can't digest it is we don't have an enzyme present here that's able to cleave the specific bond that it forms. Okay. And the details of that you don't necessarily need to know, but I do want you to know these three. Cellulose, found in plants, we cannot digest it. Starch, found in plants, we do digest it. Glycogen is how we're storing it. Okay? And each one of these things are made up of glucose only. Okay. So we'll come back to this concept. However, like I just mentioned, I'm pulling you back a little bit to understand that we need glucose to make energy. We have to make energy. And how does it do that? Well, glucose goes through a series of steps where it's constantly being broken down into smaller and smaller and smaller components. And as you're breaking it down, sometimes some of the reactions in this step are exothermic. So in essence, I take this large, complex molecule, you all have seen it, that 6 carbon, it's kind of extravagant, those single molecules, and we take it through a series of steps. And this is a very simplified version of the steps. There's about 20 or 30. And every single step, some of those reactions are endothermic. They're taking in a little bit of energy, but the most important steps in this process are exothermic. So we're taking this sugar compound and we're taking it through a series. Each one of these are chemical reactions that require an enzyme. And as we're taking it through these steps, some of those reactions are exothermic and we're making energy. This is why it's so important, and that's why it was so important to understand what these exothermic and endothermic reactions are. The exothermic was releasing large amounts of energy. And so because of that, we can use glucose to make this energy and store it. Now, this process is called glycolysis, and I do want you to know what this process is called. The steps of it we'll go into later, but glycolysis is the process of us taking a glucose compound and breaking it down into smaller and smaller and smaller components. Through each one is a chemical reaction, and we break it down to this molecule called pyruvate. Okay? And we're changing the chemical structure. Every single time we're going through a reaction, and at the end of it, we make lots and lots of energy. Okay? Because some of these steps are exothermic. Now, pyruvate then goes into another cycle where you can make more and more energy. So it just doesn't stop here. But then at the same time, now you can understand maybe why we start forming some waste products, right, in our urine, in our feces, because some of these reactions produce stuff that we don't want. It's not all perfect, right? The energy is nice and perfect, but we produce a lot of stuff that we don't want. And so we have to have a way to get rid of that, and that's where we have these disposal mechanisms. However, the most important thing is glucose can be broken down, and each step of it can produce some energy. Okay. So that was the carbohydrates. Big picture carbohydrates. It's kind of the biggest category, and then we have lipids. Okay, and lipids. I want you to think of these things as simply as fats. Okay, they are doing so many important things. One of the most important things is they form the membranes around our cells, but not just our cells. Remember, I told you that all of those things inside the cell have these membranes too. The mitochondria, the nucleus, the Golgi body, the endoplasmic reticulum. And we'll talk about each one of those components in lab too. But it's not just around the cell. Okay, they also insulate body organs. Okay, and they produce an additional storage of energy. Now, I do want you to know this, is that they form an additional storage of energy, but that energy is very efficient. Okay, lipids are also known as fats. Fats, and this is what's coming in from our diet. Fats happen to be a very, very efficient form of energy, more efficient than glucose. However, we don't use it as much because it creates some really bad byproducts. But the most important thing is that they're insoluble in water. And what do I mean by insoluble? It doesn't mix with water. Water will separate from lipids. And you'll see that time and time again. If I take water and I pour oil in it, it will separate. Okay, that's a fat. That's a lipid. They don't mix, ever. And so that's what allows our cells to separate. You remember I just told you that water's making up our body. It's making up our entire body. The membrane around the cells is made of these lipids. This is a very fluid thing, just like oil and water. What allows these cells to separate and form structure? It's the water there in our body that separates the fats around each other, and we can form these individual cells. That's what's going on. Is the fact that these lipids or these fats do not interact with water, that we can form cells. This is what causes the lipids to form around these cells. And inside our cells, that's what causes these individual things to form structure. Okay, these also contain carbon, hydrogen, oxygen. We're still talking about organic compounds, and we have three types. We have triglycerides, phospholipids, which happen to be these, and then we have steroids. Okay, and you all can take a 7-8 minute break. Just come back like 10, 11, or 10, 12, and we'll talk about this stuff. Oh yeah, I saw it yesterday. Your exam grade is already in. You took that on Thursday.
the exam you took on Thursday, but there's a homework assignment. It's a video, the concepts, the video assignments, maybe what you were talking about. And so you can make that video assignment still and turn it in to me. I would like it sooner rather than later, but the concepts are there. You just pick one concept and make like a 2-minute video or 3-minute video explaining it.
Oh, like, is it a video like teaching some?
Yeah, just like to your phone or whatever, or maybe on the PowerPoint side, and you just email it to me or text it to me. It's fine.
Okay,
Yeah, but
there
you're-
is also like a 100 marks exam. Is that we completed it?
Yeah, that's what you can do.
Okay, thank you.
Ab, how is that camera there? Is it on or? Don't touch it.
Yeah.
Okay, good. I'm going to Starbucks today. After teach lab. There's some writing done, man.
We're teaching a lab there.
No, after I get done with lab, I'll go to Starbucks.
Did you just get a drink?
No,
For what?
just get into like a writer mode. Oh shoot. What?
Great.
Ryan Davis?
Yeah, great.
I'm going way faster than I expected. Cool drive.
Are you going to the funeral tomorrow?
It's tomorrow?
Yeah.
What time?
I would have thought it would be like Friday.
What time?
Let me see. What time were we supposed to be back in class?
Is it like 10, 12? Wait, what am I
At 1 p.m.
I mean, I can make it, but I'll have to leave like right after this class. I just lost it.
Would you want to go with me or would you drive separate?
For not to drive because I have expired tags.
You know what?
My tags are expired.
Your dad might come with me so we can all. I need to get a dress and wear for it.
No, you don't have.
What am I gonna wear? Jeans?
Yeah.
I guess I could work actually.
It's Easter.
What?
It's Easter. Let's see. Okay. So we have these three types of lipids: triglycerides, phospholipids, and then steroids. First type is triglycerides. Now, again, don't forget we're talking about macromolecules. So we have individual monomers that make these things up. Okay. Those two monomers happen to be fatty acids and glycerol that are making up triglycerides. And this is what's making up what we know as fatty foods or fat foods, and then oily foods. Okay, are these triglycerides, and they provide a very efficient form of energy for our body. But again, they create some byproducts that we don't necessarily like, and cause a lot of problems. And so we don't typically like to use them as energy, although we can. Now, glycerol exists up here. It's this 3-carbon compound, and then we have a fatty acid tail. And these tails are long carbon chains. They're typically called, they form chains like this, and they're quite large. And so oftentimes when you see compounds like this, I told you that many, it wants to form 4 bonds. Oftentimes those bonds are hydrogens. Okay? And you assume that they're hydrogens bound to carbon, unless something else is written. Because don't forget, the goal is for carbon to become stable. In order for it to be stable, it needs 4 bonds. Okay, so we'll always form 4 bonds. Now, if there's nothing written, say for example on this line, you all recognize that this is carbon. There's 1 bond here, but understand that there are 4 bonds that carbon's forming here because it needs 4 to be stable. So you assume that there are 1, 2, 3 hydrogens there to form, to be stable. Now, typically the hydrogens are not written. If it was something else like oxygen, similar to what we saw with the carbohydrates, it will be written there. But in the case of this, like for example here, oxygen is written here bound to that carbon. If nothing's written there, if it's just these lines, you assume that hydrogen is bound. Carbon will always form 4 bonds. In this case, this is 1 carbon. It's bound to this other carbon. It's bound to this carbon. You have 2 hydrogens. These are cumulatively, when you have compounds that are large carbon chains with hydrogens on them, they're called hydrocarbons. Okay, hydrogen carbons. And again, what's making up the triglycerides are these large carbon chains that are bound to a glycerol. And with triglycerides, there's always 3. So that's a key thing to understand. You have glycerol, and glycerol is 3 carbons, and bound to each one of these carbons is this long hydrocarbon. You have 3 of them. That's what makes the triglyceride. Now, hopefully you all can see, and I'll say it again, is that every time you move over here, the structure is getting a little more simplified. Okay, this is the simplest way you can write it. But it's really writing this here. Okay, so that's triglycerides. And these triglycerides come in 2 forms, very important forms. One's called saturated. The other one is called unsaturated. And the saturation has to do with if you have maximum amount of hydrogens bound here or you don't. So it's the difference in how many hydrogens you have bound. If I'm looking at a tail, a triglyceride tail like this, everybody can understand this is probably where you'll have the most hydrogens bound. Okay? You will have maximum amount of hydrogens bound to carbon here. Now, if I wish you could zoom in a little bit. If I, for example, had an oxygen coming off of this carbon, you all understand that at that carbon, if there's oxygen bound like this, a double bond oxygen, there's not hydrogens bound to that bond, or that carbon. There's no hydrogens bound to that carbon. So it's taking up the space for hydrogen to bind. And so this is what I'm talking about when I'm saying the difference in maximum versus not of hydrogen bound to the carbon chain. If you have essentially a naked chain like this, this is considered a completely saturated triglyceride or a completely saturated fat. Okay? And that complete saturation allows it to have this straight chain structure.
Now say, for example, let's say I have a carbon and I add a double bond. We talked about double bond that's that's sharing of 4 electrons, right? Say we form a double bond here. So now I have this carbon that has 1, 2, 3 bonds formed to it, and you assume 1 hydrogen, right? Everybody sees that. You assume that there's 1 hydrogen here. Really, it should be 2 if it was maximally saturated. But that double bond takes up one of hydrogen's spaces. And so now we're not completely saturated with hydrogen. And so we have formed an unsaturated fat. Now, this translates to the physical structure of it. Oh, dang, I wish I had a model. I kind of have one. Let's see if I can do this. I can't. Explain with this. Now, we know that these bonds are sharing of electrons. That's all that these lines indicate. Sharing of electrons between multiple elements. Okay? Well, when you form bonds like these, you change the way that these molecules can move. Remember, I told you the name of the game is how these molecules interact with each other. At the very foundation of it, whatever thing you change in a chemical compound changes the way that it interacts with something. Okay. Well, the number of bonds has something to do with that. If I have a compound like this, and I have 1 oxygen and 1 hydrogen, you all recognize that this is 1 bond. We're only sharing 2 electrons here. Well, this 1 bond allows these molecules to rotate. Remember, we taught that, the electrons are moving, but because the electrons are moving, these molecules are spinning. They're constantly moving. Now, they're held together by the sharing of electrons, but they're constantly moving. Say I add a double bond to the picture. Here. Now, all of a sudden, I have locked these 2 elements from moving in a certain way. They can only rotate, in this case, it can only rotate, this way, or maybe this way. But these 2 elements will not be able to move. They are restricted by the sharing of electrons. That relates directly to these carbon chains. These carbons are moving, but they're not necessarily restricted. Okay, so these carbon chains are constantly moving. Well, I stick a double bond in here, all of a sudden I put a kink in the chain. Now, instead of this molecule being able to rotate freely at each one of these elements, obviously restricted by the 1 bond, all of a sudden I have now made a compound that can now only rotate a certain way. Now it's sort of rotating diagonally because it's restricted, because I put a kink in this chain. The importance of that is because when we're talking about triglycerides, we're talking about hundreds and hundreds of them, if not thousands. And they have to pack together to form what we know as oil and form butter, okay, and form all these other things within our body. Well, when you add a kink to a chain, or I have a triglyceride that's unsaturated, these are 2 interchangeable things. Unsaturated, or I'm adding a kink, or I'm adding a double bond somewhere, this changes the way that they can pack. They can't pack as tightly because all of a sudden you can see that there's a kink in this chain. I will disrupt the packing in some way. Kind of like if I'm packing a box and I put this awkward shaped thing in the box instead of just stacking a bunch of books that could evenly pack into the box. In this case, when we have a saturated fat,
There's no kinks in this chain, and they simply just stack one on top of the other. Just like if I take a box and I start stacking textbooks in them, they'll evenly fit in and they'll form a nice little full box. But what if I put some sort of awkward shape like this water bottle in there? All of a sudden those books don't stack into the box. You'll have some air space in there. They won't stack evenly in the box. And that's what's going on in this case. And that changes the composition of whatever you're trying to make. In saturated fats, where we have complete saturation of hydrogen on carbons, they stack very easily. And so you can form this solid structure, and we know this is butter. Okay? This is a very solid structure. Now, I had one kink to the chain, or maybe I had multiple. Okay? And, we add kinks to the chain, they don't stack evenly. These compounds are sort of separated, and we form a solution that we know is oil. Now, this is not water. Right? This is a very unique example of a liquid where there's no water present. This is oil. Okay? Typically, any liquid that you have, like soda, anything, blood, is made of water. But this is a liquid compound that's only oil. There's no water here. And it's because we now have kinks in the chain that don't allow them to pack evenly. And so they're dispersed. We have this oil substance. It's not solid at all. Okay? And so you can describe saturated versus unsaturated fats in the form of maximum amount of hydrogen saturation or not maximum amount of hydrogen saturation. Or you could say that there's a double bond present here and there's no double bonds present here. You could say, I would rather not use the term kinks, but you could say that there's kinks in the chain. Okay? There are multiple ways that you can describe these. But what I want you to understand is that when you change the composition or you change a chemical formula in any way, you're changing the way that it interacts with other things. And this is a prime example of that. Now, that's all I want you all to know from triglycerides. So be able to describe unsaturated versus saturated. That could be an open response question on the exam too. Know this pretty well. And we have phospholipids, okay? And these are probably the most important because these are what's making up our membrane. Phospholipids have two, I would say three. I don't have the monomers on here, but I would say three monomers. We have glycerol, fatty acid, and a phosphate group. Okay? A phosphate group is what's distinguishing triglycerides from phospholipids. We have a phosphate group that's present here on the top. Okay? Now, you can further modify these phospholipids by adding other components to them, a nitrogen-containing group, and oftentimes phospholipids do, but you can add different types of compounds that make these phospholipids a little different. Phospholipids is just a general term. However, these only have two fatty acid chains. Okay? Now, you can think of them having three chains, it's just that the third chain is that phosphate. Right? You remember on the triglyceride, this carbon was bound to another fatty acid chain. In this case, it's bound to a phosphate group that's present on the top of the carbon. Okay? Now, these molecules have a unique property. One is that they have a portion of them that likes to interact with water, and they have a portion of them that does not like to interact with water. Okay? Typically, when you have compounds that have large carbon chains like this, they don't like to interact with water. They're typically called what's called hydrophobic. They're water-fearing. And the opposite would be the phosphate heads, which are hydrophilic, or they're water-loving. Now, you can describe them in terms of their polarity, the type of bond. We talked about the covalent bonds. This fatty acid tail, when I have two carbons here, and don't look at the slide, look at me. If I have two carbons here, what type of bond would that be? Two carbons bound together. Pull from your head. What type of bond would two carbons be?
Double bond.
No, well, if we had two bonds together, it'd be a double bond, but I'm just talking about the type of bonds. Remember, we talked about ionic, we talked about hydrogen, we talked about covalent. What type of bond would two carbons be? Not ionic, because those are opposite charges. Remember, ionic is positives and negatives. Covalent bond. That's a covalent bond. And you remember we talked about two types of covalent bonds. We talked about the polar covalent bond, where we had one element that was really electronegative and one element that was not electronegative, and we formed those partial charges, like we just talked about water. And then there was the non-polar covalent bond, where the two elements had equal electronegativity. And so those electrons were being shared equally between the two elements because they each wanted electrons just as bad as the other. In the case of the polar covalent bond, the two elements, one was really electronegative and the other one was less electronegative, so the electrons were shared and stayed at the stronger element, or the more electronegative element, longer. And that's what formed those partial charges. Right? It kept the electrons away from hydrogen, or the less electronegative element, and so the hydrogen got a little bit of a partial positive, and the oxygen got a little bit of a partial negative. In the case of these phospholipids, we can describe them in their polarity, and we can describe a lot of compounds this way. And so this is a first introduction to them, but I will often tell you a lot of times when we're looking at proteins and such, I'll say that this is a polar compound or this is a non-polar compound. What's going on here are non-polar covalent bonds. You remember I told you that it was typically between two of the same elements. Well, when carbons are bound to other carbons, that is a non-polar covalent bond. There's equal sharing of electrons between them. In essence, there is no element here that's wanting the electrons more than the other. It's a very equal distribution. And that makes these things very stable. Right? There's not any tug of war between the electrons. Now, these fatty acid tails are non-polar. Okay? So we can describe them that way. You can say non-polar fatty acid tail, or you can say hydrophobic fatty acid tail. Now, I do want you to know that these terms are typically interchangeable. If I say non-polar, typically I'm indicating hydrophobic. And the same thing with polar. This phosphate head, if you can, if you can see here, we have an oxygen and a carbon. Oxygen is more electronegative than carbon. Remember, carbon was electroneutral. Oxygen is pretty electronegative. This is a polar bond. Okay? The same can be said for phosphate, although not so much, but here, certainly, a polar bond. That makes this compound, or that makes these heads hydrophilic. They like to interact with water. Now let's think about this for a second. Why would it like to interact with water? Let's pull some concepts out. Why would it like to interact with water? What is the polar bond forming? Polar bonds forming partial charges, right? You remember I showed you one of the properties of water was what? Okay, reactivity. Also, it bound to those charges. Water liked to coat around those charges. I'm telling you here, this is a polar bond. I'm forming partial charges. So doesn't that make a great place for water to want to come and bind and circulate or wrap around these charges? That's what makes it hydrophilic. That's what makes it love water. Is that we have these polar bonds here and we're forming those partial charges. And water also has partial charges. Maybe I should go back to the slide. Water has those partial charges that bind to
the negatives and positives. It switches its orientation to where the negatives interact with the positives, and then it switches its orientation where the positives interact with the negatives. It's soaking up the components. Now, this is the same thing that's occurring here. This is what makes it hydrophilic, water-loving. Water wants to bind here because of its polar properties. Does everybody see that? That's what's making it hydrophilic. Now, there are not those charges here because these are non-polar covalent bonds. You don't have any charges going on here, no partial charges. Water is wanting to stay away. It's being repelled. Now, that allows it to, does that make sense to, does that make sense to them? Understand, this is why we started way back there with the chemical component, is that they're making up everything that we understand here. And even further, this will just build and build and build on top of each other. Now, because of these two properties, you have one molecule that has two very distinct properties associated. One part that really likes water and one part that really doesn't like water. That's a unique characteristic about these phospholipids that allows them to form these layers that prevent water from getting in. And that's what forms, that's what allows us to make up these cellular membranes. And so what's going on here is that we have these heads. Now remember, this is it. I told you the first day, we'll see a lot of artists' depiction of stuff. This is still this. It just makes it easier to understand that I'm drawing the picture. But don't forget, these are all chemicals. Now, if I have these tails that don't like water, and I have these heads that do like water, what will naturally happen is that if I put it in water, they will find a way to keep water away from the parts that don't like it and let water move to the outside. And that's exactly what happens. These things form just like when you pour oil in water. So all of a sudden, all these phospholipids bind up together and form a watertight seal on each side of the, in this case, this is a cell. On each side of the cell, the inside and the outside, a watertight seal that keeps water from coming in here. Because this is hydrophobic. Because these are hydrophobic, they form this seal that keeps water out. And that allows us to form a nice layer that separates components. This is what's forming the outside of the cell. It makes a whole circular structure that forms around the outside of the cell because our body is water. And so it's just like pouring oil in water. These things will just form because these tails do not like to interact with water. Okay. Now, hopefully you'll understand, again, these are making the outside of the cell. And I keep saying, just like oil and water, these things are very fluid. Okay? They're very wavy because, water's moving and we're constantly moving. These things are very fluid with water. Just like oil and water. This is the same thing. This is essentially oil. Okay? They're lipids, they're fats. Now, that leads us to this structure, it's steroids. And steroids are an interesting type of lipid. They always have this ring structure. The rest of this, it's kind of different depending on what we're talking about, but they always have these 4 hydrocarbon rings. Always. I will give you a picture of this and tell you to tell me what it is. You need to be able to identify this because these make up a lot of compounds. These are steroid compounds, but they're still lipids. They have a 4-ring structure associated with them, and the most important one is cholesterol. And why is it so important is I just told you that these lipid membranes, what's being formed here, is very fluid. Very, very fluid. Because water's surrounding it, water's on the inside of it, it's just like oil. Well, steroids will embed themselves on the outside of this membrane in between these lipids.
those molecules that you just saw will embed themselves into the membrane. And that will allow it to have a little bit more rigidity. So it doesn't break off as easily. It will allow it to be less fluid, and so we have more structure associated with the cell. Okay, that's why steroids are very important, specifically cholesterol. Cholesterol is doing this. Now, one thing that you'll see next class is that cholesterol is also incredibly important because it's a precursor to a lot of hormones in our body. Some of the most important are testosterone, estrogen, cortisol, which is stress hormone, aldosterone. There's more there, and there's many, many others. But cholesterol is the first step. And so, like, these are all steroids. And so when we want to make these steroids in our body, at whatever organ, whatever endocrine organ it is, when we want to make these compounds, we start with cholesterol. And then we have one enzymatic step that makes it into something different, and another enzymatic step that makes it into something different. And eventually some of those steps will make each one of these. So each one of these compounds are made from the same molecule, cholesterol. And they go through multiple steps to get there. Okay? So you make estrogen from the same thing that you make testosterone from. All that matters is the enzymes that you have present in that cell. So you can have multiple enzymes that take it all the way to estrogen. Or you can take away one that will only keep it at testosterone. Or the same for cortisol or aldosterone, okay? So, but understand that every single one of these components though, every single one of these hormones, along with vitamin D and of course cholesterol, all have the same 4-ring structure, and that's right here because they're steroids. Okay, when I say steroids, immediately you should think of this figure here. What gives it the difference is what you add on to these portions, to some of these carbons, and typically it's right here. What you add on to it gives it the different hormone or maybe vitamin D, and again, it's changing the chemical component of it that allows it to interact differently with other stuff. And that's why we have these different things. But understand they look very similar. Okay. I would like you to know the function of cholesterol though, is that it's maintaining rigidity in the membrane. Okay. And the last one, we'll spend some time on this, and we'll probably start off Tuesday talking about this.